Hi everyone, and thank you for the opportunity to present here. I'm Riccardo Calandrelli from the Sheng Lab at University of California, San Diego, and I'm happy to spend the next 3 minutes to present our latest work where we leveraged RNA-chromatin interactions to study the mechanisms behind endothelial cell dysfunction. So endothelial cells are the most inside monolayer of cells in the lumen of vessels, and they are subjected to all the detrimental factors present in blood in many diseases, like for example in diabetes, which can provoke EC dysfunction by providing mainly two types of stresses, high blood glucose level and inflammation. So we wonder how these factors lead to endothelial cell dysfunction. And in order to answer this question, we established an in vitro system using UVEC to mimic the temporal progression from healthy to diabetic state by generating 3 samples at different time points or disease stages. An healthy condition, also named day 0, our control with untreated cells, a pre-diabetic condition where cells were treated with high glucose and TNF-alpha for 3 days, and finally a diabetic condition where cells were subjected to the same treatment for 7 days. Then we used a combination of 3 techniques, single-cell RNA sequencing, in situ IC and IMARCI, to characterize the temporal changes in transcriptome, chromatin interactions, and RNA-chromatin interactions. In addition, we used also single-cell RNA sequencing from in vivo vascular endothelial cells from healthy and diabetic donors for validation. So first, we explored the single-cell RNA sequencing to confirm that our model was indeed reproducing EC dysfunction. And indeed, our 3 samples were well separated in the Disney plot, and also we saw that genes contributing to EC dysfunction were upregulating in the 3 and the 7 samples. Then we looked at the genome interactome, and while chromatin interactions from IC weren't really changing over the time course, we noticed instead a strong increase in the number of inter-chromosomal RNA-DNA interactions, here shown in blue, meaning the amount of RNAs in proximity to a piece of DNA on a different chromosome. Moreover, we also observed that a lot of these chromatin-associated RNAs were transcribed from and targeting super-enhancers, as you can see here. So we thought that changes on gene expression in dysfunctional endothelial cells could be caused by inter-chromosomal RNA-DNA interactions between super-enhancers. Indeed, if we represent super-enhancers in interaction networks, you can clearly see how the number of interactions increases over the time course, and also how several central hubs that become active in the 3 and the 7 are super-enhancers overlapping critical genes in EC dysfunction, as we labeled here. Then we focused on the novel interaction established by the 7 between 2 super-enhancers overlapping SERPIN1 and Link607, as pointed here by the blue arrow. And strikingly, we also observed that these 2 genes should also increase co-expression in dysfunctional single cells compared to the healthy one, both in vitro and in vivo. In the interest of time, I must conclude here, and I would like just to mention that this project was carried out in collaboration with the Jen Bowman Chen Lab at City of Hope, and I would like in particular to acknowledge Lixia and Hinjun, who were behind the majority of the biological experiment, in addition to other members and my PI, Dr. Zhong. Also, you can find here the reference to the paper, and see you at poster number 36. Thank you.